Okay, so once you see this, what happens? You can see that the column will be packed with the stationary phase, which can also called as like matrix, and we can also have like a mobile phase, so which will be placed at the top of the column, and then which will come down to the bottom, right? So when we place the sample molecules here, you see there are a layer of sample molecules at the top of the packed column, and then when we place the solvent or like what we can say like mobile phase, then these molecules will be move along with the mobile phase. So they will move along with that. So that's why in the second one, you can see the molecules. There are two types of molecules you can see here, like a blue colored and then blue colored squares, and then like brown color triangles. And you see this blue color squares having ligands in the matrix. So that's why they can get attached to that particular matrix. And whereas this triangle brown color molecules doesn't have any binding capacity here in the matrix. So that's why what happens, they will be released, they will be moved along with the solvent, or you can say like mobile phase. Okay, so after this, what happens? Only the molecules which are having specific binding site, so they bind here, and the remaining molecules will get separated. So then afterwards, what happens? We'll change the conditions of the column in order to release the bounded molecules which are already bounded here. They will be released in the next step. So in total, you can see the chromatography will follow the same procedure, like first of all, packing of column, and then selection of the solvent or like mobile phase, and then adding of sample or loading of sample on the top of the column, and then adding the mobile phase and then running the mobile phase. So based on that, what happens, the molecules will get separate, and some molecules will get attached, so will be there uh, inside the matrix, and some molecules will get elute. Okay, and the molecules which are already attached may be separated by using a different type of pH or something like that, what I told you in the ion exchange chromatography. It's the same kind of thing. And some methods may not be having like binding capacities, and they may be having a different kind of capacity. Or you can also say the gel permeation chromatography will have a different kind of capacity, how it gets separate. You will not be having like a third step, you'll have only second step, where we can see the molecules will get separated based on their molecular size. So slowly, so each fraction will have a different molecule, so that we can see. But in case of affinity chromatography, you see this is agar bead. Agar bead is like, the rounded ones here in this picture, right? This matrix, I can say. And this matrix, so usually can be added with a ligand. So this is a ligand here. You see, this ligand is added to the agarose bead, and the ligand here it is mentioned as like insulin molecule will be added to this. And then once we add the sample molecules on top of it, and when they are moving, so when they are near to this ligands, and then you see the protein to be purified should be attached to this insulin receptor. So the protein should be purified, should be the insulin receptor. So we are going to separate the insulin receptor. So when the insulin receptor is going to separate, when the insulin molecule is there, insulin molecule and the insulin receptor will have a binding capacity, so that it forms as a complex. Receptor, insulin and insulin receptor complex will be formed. So that means we are adding, what we are adding? We are adding a ligand molecule, so that ligand molecule will have the affinity towards the molecule which we are going to separate. So the molecule which we are going to separate here is insulin receptor. So that insulin receptor, as and when it is passing through these beads and which are attached with the insulin molecule, so these molecules will get attached. So that means insulin receptor molecules will get attached to the insulin molecule, which is acting as a ligand, which is attached to the stationary phase, right? So this is how what happens. They will be folded here, and the remaining all molecules will get eluted. So along with the mobile phase. And whereas, so once it is eluted, and then you can see that the column will be subjected for the extraction of the desired molecule. So especially in this case, it is insulin receptor. right? That is how we can isolate. So now you can have a basic idea about what is affinity chromatography. So the affinity is having towards the ligand. So the biological molecule is having affinity towards the ligand. So the ligand here, it is a insulin molecule, and the ligand molecules will be get changed each and every time. It's all depends on based on what kind of molecule you are going to separate. So it is very specific. The ligand is specific, and as at the same time, the molecule which we are going to separate is also specific. So such a way that we can isolate the molecules. Okay, so when we see the introduction here, so what is affinity chromatography? So because I already told you what is affinity chromatography. So that chromatography is a method of separation, separating the biochemical mixtures based on high specific interaction such as that between antigen, antibody, enzyme, substrate. That means you see antigen, antibody. Antigen will act as a ligand, or antibody will act as a ligand, and we are going to separate the other molecule. So if it is antigen as a ligand, and we are going to separate the antigen antibody, right? So the antibody will be separated. Similarly, the enzyme and substrate. If the substrate will act as a, when it acts as a, usually a substrate will act as a ligand. So when it is acts as a ligand, and when we add enzyme, so the enzyme will go and bind to it. So what will happen automatically? The enzyme molecule will be get attached with this substrate molecule. So we will have ligand molecules, and which are specifically binds to the molecule which we are going to separate. Or you can also like receptor and its related a molecule, protein molecule or any other molecule. So that also we can use and we can separate. So basically the affinity chromatography is involved in separation of biological molecules using the ligands for that particular or specific type of molecules that we can see. And you see what is the principle involved in the affinity chromatography. So in the affinity chromatography, we make a matrix which we call it as like a stationary phase, and usually it may be like a saccharose, agarose, or something like that. We'll see what are the compounds which can be used in making the affinity chromatography, or like columns preparation. That those things we'll see. So the matrix is a stationary phase, will be fixed, and then for that we are going to add ligand molecules. So the ligand molecules will have an attachment, like ligand can have the capacity that it can attach it to the matrix molecule. It can also have the capacity to attach with the molecules which is going to be separated, or you can call it as like the molecule of our interest. Okay, so the ligand can be selected based on what kind of molecule is going to be separated. So if you separate that, and you can also select this ligand should be added to this matrix. That means they should have a compatibility, that the matrix molecule should have compatibility with the ligand so that it can get attached. And then the ligand should have a compatibility with the molecule of our interest, right? So that is how we can separate the molecules. Okay, so let's see. Well, in the first reaction, what happens? The saccharose is a material. Saccharose is a material, and this material will be used as a matrix molecule or stationary phase, and this will be added with a ligand molecule, and this ligand molecule will form like this. It forms as a complex like immobilized ligand complex. Right now the ligand molecule cannot move, and in the next step the immobilized ligand molecule is exposed to the sample molecule. So the sample molecule, in the sense the sample molecule is having a many molecules, including like the non-specific molecules and the specific molecules. Specific molecules is always having the capacity to bind with the ligand. So the ligand can be added with this, so that what happens, it will get attached, and so that what will happen, you can see the matrix, ligand, and then molecule complex. Molecule or the ligand, sorry, the matrix, ligand, a sample complex will form. So it's a complex. But you see, the other molecules which are non-specific to this ligand will get eluted. So they will be removed. They will be removed. Then finally, what happens? We have to extract our sample. So how do we extract our sample? So this complex this complex will be exposed to the other molecules, or like you can change the pH, or you can also add some other molecules which are having more affinity towards this ligand than the sample molecule. So in that case, when you change the pH, then automatically the binding between the ligand molecule and the sample molecule will get reduced, or you can also say that weaken. So when it is weakened, automatically the sample molecules will get dissociated from the ligand molecule. See, that is one way that you can do it. Like you can change the pH and then you can do that. You can have it, and you can also see. You can also have like we can also add some molecules like which are having more affinity towards this ligand. So when we add the more affinity molecules of the ligand, then automatically ligand will dissociate from the sample molecules, and then these sample molecules will be dissociated, and the other molecules which are having high affinity, so they will get attached to this. So they will have a more bonding, affinity bonding than the sample molecules. But our aim is to isolate only the sample molecules. So we may isolate the sample molecule separately, right? This is what we can see. The basic principle involved here is like immobilized ligand can have the capacity to separate the specific molecules, okay, and elute the impurities. And from that, you can elute the specific molecules from the ligand attachment, so that you can isolate the, purify the, or you get the pure compound from this method, right? Okay, so what is the different characteristic properties which we can see with this ideal matrix? So what kind of characteristics it should have? So if you see, it must be stable during binding of macromolecules and in subsequent elutions. So that means it should be stable. It should not be changed its chemical property. It should not be changed its physical property. What does it mean by its chemical property and physical property? So when it changes its chemical property, then automatically the ligand molecules will get some, separated from the matrix molecule. So that's why what happens, we have to be very sure that the uh, uh, stationary phase which we are going to use, or you can also see like the matrix which we are going to use should be having stable during the binding of macromolecules. So that means when it binds, it should not interact with the molecules, macromolecules. So for example, if there is an enzyme will attach to the ligand molecule, and then the ligand molecule enzyme will not interact with the it should not interact with the matrix. So the matrix, we can simply say that it is an inert one. So the inert, because it is inert, so then it won't be interact with any other molecule. So that we can see that that should be the stable characteristic property of the matrix molecule, and also it should not be changed any. It should not have any change in the subsequent elements. What does it mean by subsequent elutions? Because I told you while elution, we can have two methods. That is, one is like you can change the pH, and then when we change the pH, and then you can have a different kind of like the interaction between the ligand and the sample molecules will be get dissociated. Then automatically the sample molecule will be released from the matrix, right? Or you can say like from ligand. But when the matrix molecule is not stable at this pH. That means you are going to add either acid or pH, acid or like base. Depends on what kind of pH you need for the elution of the ligand binded molecules, right? So in that case, when you add some acid or base to change the pH of the column, then automatically you can see that the matrix should not get affected. So the matrix should be stable in that particular pH. So that's why while selecting the matrix, you have to be very sure that you are going to separate certain molecule, and then you have to select the certain type of matrix. And you should not use any type of matrix for any type of molecules, right? So that is the one which we can see. And then it must be at the most interactive with a specific compound and only weakly uh, with other molecules to minimize a non-specific absorption. So that means when we see ligands, we know that ligands will be added to this matrix. So the matrix should be having only a specific interaction with the specific ligand, but not with any other molecule. So the non-specific molecule absorption should not be happen. So only the specific molecule absorption should happen. So that's why what happens, the ligand matrix complex should be stable. It should not be interact, or it should not be a non-specific. When it is non-specific, when we add some molecules, then again what will happen? So this a matrix can also form a ligand bonding with this particular molecule. So when it forms, and then you cannot say that it is having only a single type of ligand. You can say that it is having multiple types of ligands. Okay, then we won't be able to separate our molecules, because the different molecules, different ligands will show the different specificity, so that they will have a different type of molecule attachment, so that we will see that the number of molecules, or you can say a number of species of molecules, will be attached in the matrix. So that we will see. So that should be avoided, right? So, and then it should exhibit good flow properties. It should have a good flow property. So that means when we pack the uh, ligands, or you can say that when we pack this beads and all, like agarose beads or like saffrose beads, and so then they should not close completely. So that means when we place some what we call it as like mobile phase on top of it, and then it should allow the movement of that mobile phase from top to the bottom. It should not be completely packed and without passing any material. So it should allow the passage of the molecules, and you can also see that the flow properties. It should exhibit the flow properties, right? So that means it should be macroporous. When it is macroporous, then it allows the large molecules to access and binding site, and need to have the good flow properties. So if when it has good flow properties, and then it's more easy to isolate the molecules without any contamination. So that's why you have to be sure that you are selecting the matrix which is having a good flow properties. Then it should have, it should avoid a non-specific absorption. So the matrix molecule should not be having any interaction with our sample molecule. So that means the sample will have a different kind of molecules, right? So it's a mixture of molecules we have in the sample. And when we want to isolate only a specific molecule, then you have to go for the affinity chromatography. When we go for affinity chromatography, there you can see like only ligands will bind only to the specific molecules, and but not the non-specific molecules. See, for example, if the matrix molecule is having some other groups other than this ligand molecule, and if that specific groups are like different groups which are present or located on the matrix molecule, if they interact with the other sample molecules, not the molecule which we need. So it can be binded only with the specific ligand, but the other molecules which are there in the sample, they if they interact with the matrix molecule, they may also be stay, or you can also say like they may also bound with the matrix molecule. So that non-specific binding should not be happened. So you have to be very sure that when you are going for selecting the sample for separation, you have to be very sure that the matrix molecule may not be having any non-specific interaction, or the crossing interactions with the sample molecules which we are going to allow to pass through the sample. Okay, so this is most important aspect which we can see with this uh, ideal characters of the matrix. But you can see it has its own kind of drawbacks, and not all of them will have all kinds of characters, but you can see only a few of them will have the good characters. Okay, so what are the different types of matrix materials can be used? What materials can be used to prepare the affinity chromatography column? So to prepare the affinity chromatography column, so we have different types of matrix materials available, and those are like dextrons, porous glasses, polyacrylamides, agarose, and then cellulose. So all these molecules will act as, all these chemicals will act as making the column, or you can also say like a matrix, right? Or stationary phase. So dextran is a polysaccharide and porous glass is a glass material, so which can have the adhering capacity with the ligand molecules. So a small glass wool and తెలుసు కదా మనకి, you know that, right? The glass wool. So glass wool is very specific one where we can see this glass wool can get packed in the column. So when it get packed in the column, then it will have some pores. It makes some pores, right? And this glass wool can help us to make the porous glass wool can help us to make the column, or it will act as a matrix. Then polyacrylamide gels. We already know. We saw this sodium dodecyl sulfate polyacrylamide gel electrophoresis we have discussed in the earlier classes, right? So when we talk about the polyacrylamide gel, there I told you the polyacrylamide gels are always having pore, right? Pores it has. Then when we apply the electricity and the buffer is moving, then along with the buffer based on their molecular size, they will move along with the buffer. So the porosity will also make the passage of these molecules, and so that they will get separated. So then separated protein molecules will be isolated in the gel electrophoresis, or especially in the polyacrylamide gel electrophoresis. But you see, the polyacrylamide, since it has the capacity to make the porosity containing gel, so that's why it can be packed inside the. So it may be a syringe, or it may be a burette, or it may be anything else, which we can call it as like a column. So in that column, we are going to pack it. Okay, so once we pack them, then you can, what we can say, like you can add the sample on the top of it and then see how the molecules are moving. So it's all depends on a porosity will also help to separate the molecules. That is, large molecules will stay in the top, and then medium sized molecules will stay in the middle, and then whereas small molecules will be in the bottom, right? So this is what we will observe with the polyacrylamide gels. Then you can also see agarose. Agarose is prepared from a particular material, which we call it as like agarose. Sorry, the agar. Okay, so the agar is the organism. Agar is the algae. So it is an algae. From there we can extract the agar, and agar can be used to purify, and the purified agar can be called as agarose. So you can make agarose. Agarose can also be used in making the gels. Especially it can be used for making uh, gels for the isolation of DNA molecules. That means nucleic acid isolations. That we can see with this agarose gel. Okay, agarose gel is very specific. It is a molecular grade chemical, and this chemical can be used to prepare the uh, what we can say like a matrix. Okay, in case of affinity chromatography. Cellulose is another material. Cellulose is also a polysaccharide. So all these polysaccharides, when we heat them, so these polysaccharides will form as a gel. Okay, so it may be agarose gel, it may be polyacrylamide gel, it may be dextran, or it may be cellulose. All of them should be heated, and then they will be get packed into a particular porous material. That particular porous material is sufficient for isolating the molecules in the affinity chromatography. So all of them will be packed in the affinity columns. So what are the different properties which we can see with this ligand and the matrix? So what are attributed characters properties for this two molecules? The ligand is covalently attached to the supporting matrix so that the chromatography materials can be designed for a specific purification. You can see the ligand is covalently attached to the supporting matrix, and so that the chromatography materials can be designed for a specific purification. That means, what does it mean? So when the purification is required for the proteins, when the purification is required for the other materials, and then other materials, in that case, you can see you have to be very specific that the matrix material, selection of the matrix material, and also selection of the ligand molecule. So the ligand molecule selection and the matrix material is always interlinked. So you have to be very sure that they are not what we can say uh, they are not having the compatibility. They should have compatibility. So we have to select the compatible solid material, or you can say like matrix material, and also ligand. The chemical nature of the ligand is denatured by biological specificity. So usually the ligand is having the chemical nature, and let me give you examples also. So then the of the compound, so that to re-separate it. So usually what happens when we have a ligand molecule, see for example, if the ligand molecule is a substrate molecule. So when the substrate molecule is there, you just imagine one. When the substrate molecule is there, and then automatically when we add substrate is there, then we are adding enzyme molecule to separate. So when the enzyme molecule encounters the substrate molecule, then automatically the enzyme will go and bind to the substrate. What is the reaction will going on with when the enzyme binds with the substrate molecule? You tell me. When the substrate molecule is attached to the enzyme, what will happen? Anybody?
Enzyme substrate reaction will be occur, sir.
So the enzyme, when it reacts with the substrate molecule, so it leads to the formation of product. So enzymatic reaction will take, sir, which leads to the formation of products. Is it not? Right? So when the ligand is a biologically specific, and with the chemical nature of ligand is determined by the biological specificity of the compound, and two are re-separated. So we have to be very sure that this molecule is not separated because of the conversion of the ligand molecule into the uh, products. Okay, so the chemical nature of ligand is determined by biological specificity of the compound, and to re-separate it so that you can use it. See, for example, if the ligand is attached to that enzyme, that means enzyme, then automatically what happens, the other molecules will go down. But you can see that the enzyme can also be converts this particular substrate molecule into product. So that's why when the product molecule is formed, then the enzyme molecule will be get released from the ligand. So when the ligand is free of the molecules, then automatically you can see there is a specific, what we can say. So the release of the molecules will happen. So you don't need to change the pH, or you don't need to change any affinity molecule towards that. Okay. And for example, in case of enzyme purification, the ligand attached to matrix is usually powerful inhibition, which will bind only one enzyme in a complex condition. So we use like a powerful inhibitor, and which will bind only to the enzyme, so the specific enzyme, but not to the complex of the molecules, right, which are present in the sample. That we can say. And then while selecting ligand, one should consider one should consider the selective ligand will bind one particular compound. So compound to be separated. So the compound which we are going to separate should be having affinity towards the ligand, but not all other molecules. So we are going to add as a crude or like isolated sample on the top of the column. And then the ligand should have only a specific binding for the specific molecule, or you can say like the molecule of our interest. And it should be binded, but not all the other molecules. So when all other molecules are bounded, then automatically you can see non-specific molecule separation will happen. This will take. So while selecting ligand, we should be very careful. We have to consider that the selective ligand will bind only one particular compound, but ligand should be very specific. Okay. So the compound to be separated should be binded with this particular ligand molecule. Then it is essential that the ligand should possess the suitable chemical group that will be involved in the binding of ligand to the macromolecules to be separated. So that means it is essential that the ligand should possess suitable chemical group So it should have a suitable chemical group which is involved in the binding of ligand molecule and the separation of the macromolecules. That means what will happen? So first we'll make the matrix. So the matrix is having some kind of stationary phase. Then you have to add the ligand. So that means when we add the ligand, and the ligand should be attached to the internal tissues of the person, or internal matrix of the column, and then automatically what happens? The other molecules, or you can say that the macromolecules which are specific to the ligand, will be binded during the separation process. But while packing, it is essential that you have to remember the matrix molecule should have some specific groups to interact with the ligand so that it can have a covalent bonding, right? That's what we can say. And then ligand should possess functional groups that will bind with the matrix. So ligand should have some groups, some groups, or you can say like functional groups, which can have a binding with the matrix, because matrix already having some group. But this matrix, whatever the group which is having by the matrix, should also be binded with the ligand molecule. So that means the ligand should also have some other group which can interact with this particular matrix groups. So then it forms as a stable ligand, a matrix complex will form. Then the most common such groups are amine groups, carboxylic groups, phenolic groups, and so on. So what will happen? So the groups which require for the attachment of this particular ligand and the matrix molecule is usually amine groups it should have, so that the amine groups in both the groups can be attached one to other. Carboxylic groups can also help, so the carboxylic groups can be used to attach the ligand and the matrix molecule. And then phenolic groups can also be make the bonding between the matrix molecule and the ligand molecule. And then alcoholic groups. So these will also have an important role in what we call it as, like, they can be used in separation of various molecules, where you can see the phenolic groups can be used for especially for the protein molecules, because the proteins can interact with the phenol. And alcoholic groups can also be used for separating the certain groups of the compounds which are involved in uh, or we can say that which are involved in the secondary regulation of the plants or any other metabolic reactions. And how do we select the gel or ligand? How do we select them? So the selection of a gel or ligand, so usually it's all purely depends on what kind of molecule you are going to separate. So if you need any gel or a ligand, so you have to make sure that's what kind of molecule you are going to separate. So the molecule which we are going to separate always plays an important role in selecting the matrix and also ligand. So the matrix and ligand is always depends on the molecule to be separated. So the many type of matrix ligand systems are available, and commercially available they are, and then the costs are reasonable, so that's why you can buy them. And so time can be saved by purchasing pre-active activated gel for direct attachment of the ligand. So that means in the commercial products, or you can also say from the companies, you can also get the packed columns, but these packed columns should be activated. So once they get activated, then you can use them for isolating the molecules. Okay, is it clear, ma?
Yes, sir.
So is
Yes,
everyone
sir.
clear with this?
Yes, sir.
Yes, sir.
Okay, so what are the different types of commonly used ligands which you can in the preparation of matrix, and the affinity molecules which we are going to use, that means ligands which we are going to use to separate different molecules isolations, right? So the affinity molecules is usually they are the molecules which can be get separated from the matrix. Okay, let's see different types here, like nucleotides. See, nucleotides. So especially the nucleotides like 5 prime adenosine monophosphate or 2,5 adenosine monophosphate. So that means at the 2th position, and then 5th position you can see there is uh, like phosphate group and linkage. So these two nucleotides usually AMP 5-AMP can act as NAD dependent. That means it can be used and to separate a specific molecule called as like NAD plus dependent NAD dependent dehydrogenase enzyme and smoke kinase enzymes. Okay, so the NAD dependent enzymes can be isolated, can be separated by using affinity chromatography when we use like nucleotides like AMP, adenosine monophosphate, as a molecule for their separation, ligand for their separation. And similarly, you can also see that like a 2,5-AMP is another molecule which can also separate the enzymes which are involved in NADP plus dependent enzymes. So that the NADP plus dependent enzymes can be removed, and so that what will happen, these enzymes will get separated, right? So when we use like a T25 AMP as a ligand in the matrix, then fatty acids can be used as separation of this molecule like ligands, and their ligands they can be binded with this protein molecule so that you can see lipoproteins will get separated. So they are proteins, but they can be separated by the what we can call it as. So what we can call them as. Why Bashir is going to lobby all the time? Lobby. Okay, so fatty acids can be separated. But Bashir,
Yes, sir.
what happened?
Nothing, sir.
Yeah, okay. So the fatty acids can be separated. So sorry, the fatty acids will be used as a ligand and can be separated with the molecules like fatty acid binding proteins. So they can be easily isolated. So these binding proteins will always help us to know the biological functions of these molecules, how they can perform the function and molecular mechanism of the binding and then preparation of any other molecule that we will see. Then soybean, sorry, the protein A and protein G can also be used as a ligands. And usually this protein A and protein G are especially required for isolation of immunoglobulins because they are anti-immunoglobulin molecules. So that's why what happens, the anti-immunoglobulin molecules will get attached to this protein A or protein G ligands, and so that you can isolate the immunoglobulins. And similarly, the soybean lectin is another molecule which can be used as a ligand, but it is used in separation of like glycoprotein containing N-acetyl N-acetyl micro N-acetyl micro or DB galacto pyrobosyl residues. Galactopyrobosyl residues. So these residues are like N-acetyl residues can be glycoprotein containing molecules, so they can be easily separated by using the soybean lectin, right? And the next is phenyl boronate. Phenyl boronate is another chemical which can be acted as ligand, and then it can be used to prepare or to isolate a glycoprotein molecule. So only the glycoproteins can get separated from the phenyl boronate ligand usage. Okay. The next is polyatyl. So we can also use polyatyl. So the polyatyl is very specific that it can be attached with or it can be used to separate specific RNA molecules which are having a poly sequence, a run of poly sequence. So the poly sequence is very common in case of mRNA molecules. So the mRNA molecule will have a different kind of composition, like it has 5 prime capping, and then it has 3 prime polyatyl, and Other process which can be happen here is like a splicing. So the splicing can happen, and so that the molecule will become into the mature molecule, and the mature molecule is always holding a run of poly because it doesn't have any thymine molecule. So that's why what happens, it will be having uracil molecules. So the uracil is always complementary with adenosine. So it is complementary with the adenosine molecule. So when it is complementary with the adenosine molecule, and naturally the RNA molecules are having a enough poly U at its tail, then automatically what happens? This poly U, what we can say like a poly A tail, which is attached in the matrix, will have the capacity to attach with this uh, poly poly-U run. So when the poly-U will get attached, then you can see that the poly-A can also get attached, and so that it forms, then RNA molecules will be isolated. That means the remaining molecules will get isolated, and then we'll go for the RNA molecule to L state. We'll get the total pure mRNA molecule with this, right? And I'll explain you how the poly-A tail and affinity chromatography will take some. Okay. And then the next is lysine molecule. So we can also use lysine as a ligand molecule for isolation of rRNA. So the rRNA molecule is always isolated by using lysine as a ligand. Okay, so these are the different molecules which we can see. Okay, so maybe I can stop here. Friends, it's already like 12:58. Is anybody having any doubt in up to now? Any doubt?
No,
So
sir.
who is talking? Amravati. Amruta, sorry. But Amruta, what are you doing? Okay, so if you don't have any doubts in this, then I'm going to